From Microbe TV, this is Office Hours, and today is Wednesday, July 26, 2023. I'm Vincent Racaniello, and joining me tonight from Tacoma, Washington, Mark Martin. Where is Mark? Where did he go? Look at that. I thought I got it perfect. Hey, Mark. Welcome.
It's nice to see you, Vincent.
Welcome to Office Hours. You know all about office hours because you're a professor and you hold your own office hours, right?
I do, but I'm guessing the attendance here will be better than at actual office hours. That's been a problem in recent years. I used to use bribery, bring candies or snacks. You think that would help?
No, doesn't help. Now, Brienne does that too. She gives food, but when I do, I even do my office hours on Zoom for the class, and maybe 5, 6 people show up, but at least someone shows up every week. But this one, we have had a few hundred people here, and they wouldn't fit in my office, so this is the right way to go, right?
I think it's great.
But it's an interesting concept that you do your office hours for the world, because anyone in the world could sign in. And welcome, world, welcome everyone to another installment. I want to thank our moderators at the start here. We have Les tonight. We have Andrew. We have Barb Mack. We have Tom. I believe that's it for now. Thank you so much for being here and keeping things civilized, shall we say? And Mark, you're in Tacoma, Washington, correct?
I am in Tacoma, Washington, and now I can see all the people making comments over there.
Yeah, there. If you've got the YouTube window open, hopefully you have the sound off so you don't get an echo. That you can see the people and where they're from. Tell Mark where you're from. I know that Les, our moderator, is from California, and Andrew is from New Zealand. Barb Mack is from the UK. Tom could be either in Oregon or Wisconsin. He sometimes is in both. And here, Silvio Pina is from Burbank, California, I guess. Western Massachusetts. Paris, Texas. Oh my gosh, there was a movie called Paris, Texas, Fox P3. Did you see it? It was quite an interesting movie. I saw it years ago. Brazil, Rafael is from Brazil. Who else do we have here? Guatemala. Wow. I want to see the Scandinavians, because I know there's some people from Scandinavia who lurk. The Brazilian Amazon. Hot and dry. Toledo, Ohio. Inner London. Oh, here's Peak. Peak is one of our mods who just graduated high school. He's going to UCLA in the fall. And he's, I don't know where he is. Santa Barbara. Tom is in the Oregon Coast Range right now. Not too far from you, Mark, right? Oregon?
Well, it's a little south, but it's really pretty down there.
It's closer. Peak is from California. Very good. Richard is from Santa Barbara. Oh, Afterbobble is from Bologna, Italy. Here's H. Zhu from Austin, Texas. Hui Ping. And John knows how to say Washington and not Washington State. I like if you just say Washington. Mark, you just say Washington. You don't say Washington State, do you?
I just say Washington. I was born and raised in California, so I'm used to that. We used to say Washington State all the time, but I quickly learned not to say that. Vincent, you know, when I moved up here and I didn't really know anybody, and a next door neighbor came over and said that they'd spent the weekend in Leavenworth. To me, that's the big correctional facility in Kansas.
Yes.
But here in Washington, it's a fake Bavarian town, which is lovely.
Fake barbarian town. That's great. Northern Alberta, Canada, Philadelphia, Portland, Oregon. Here's Kip. 2 Farm Ds from San Francisco. Kip was just at the incubator a couple of weeks ago. Alina's from Coxsackie, New York, which I really like, Mark, because that's the town upstate that the virus was named after. Alina sent me an email so we could get you that microscope. Vincent at microbe.tv. Martha's from Charleston, South Carolina, the home of Michael Schmidt. Pismo Beach, California. Dutchess County, New York. New Zealand. Another New Zealand. We have 2 New Zealanders now. Atlanta, Boulder, Colorado, Iowa. Don't you love it? Hey, Rima. Rima with her lovely cat there. Singapore again. Hello again. That's quite good. I often say Washington for D.C., just saying. We could say whatever you want. Some people just say D.C.
You know, Vincent, there's a student of mine from a million years ago who's on here watching. Hello, Anne. Anne Rice.
Wow. Anne Rice, the writer?
No.
Because I know you were a writer, so you might know Anne Rice.
I had lots of email exchanges, seriously, with Anne Rice. She was a very lovely lady.
The one with the vampire series, right? Interview with the Vampire.
Yes.
Which some were better than others, you know.
Mm-hmm. Isn't that true for most things?
Yeah. Chris is from Montreal. India. There we go. That's far away. Sperryville, Virginia. Renton, Washington. Do you know Renton, Washington?
I do. Right up the Interstate 5.
Aren't we supposed to talk about the weather? It's 97 degrees. I don't know what it is here. It's not that hot. Is it hot out there in Tacoma?
Oh, no. It's really nice today. It's been a little bit warm, but it's like in the low 80s right now. So it's nice.
Toronto, Ontario, South Jordan, Utah. And Patricia says, would love to see one of my former professors on here. Well, aren't I your professor? Aren't I everyone's virology professor? Here's Anne Rice, Mark. Say hi. Not the same Anne, but it's a good name.
She has so many cool stories about when I first started teaching.
Let's hear your story. Where are you from originally?
Me?
Yeah.
I was born in Compton, California, which everyone's impressed by.
Why is that?
Well, there was a rap group, one of the first gangster rap groups called NWA, and their first album was called Straight Outta Compton. Oh. And in fact, I have the birth certificate to prove that I'm straight out of Compton. I was raised in North Long Beach in Southern California. And then from there, I went on to UCLA for my undergraduate work. I went to Stanford for my PhD. I have no idea how I got in there, but I'm glad I did. I did some postdoctoral work at UC San Diego. And then I worked in biotech for about 10 years and got back to academia.
Well, now, you used to write fiction, right?
I did.
When did that come in?
Well, I like science fiction. I've read a lot of it. I like science fiction, and I'm not as much of a fan of fantasy. It's it's just a preference thing. I have no problem with it. And I got to know several of the science fiction writers in the Southern California community. Gregory Benford is one of my best friends. David Brin, Werner Vinge. And as a result, someone said I'm OG. Yes, the original germ. That's what my students actually called me once. But it's interesting because I got to know them all. I met George Martin before he got really famous with Game of Thrones, that kind of stuff. So I would hang out with these folks. And then I spent a lot of time with Greg Benford talking about his fiction and going over things. And then he invited me to collaborate on a couple of novels with him, which I did. And it was at a time where I'd been laid off from my biotech job. And people think you make a lot of money when you write fiction. No, some people do. I am not one of them. but at least it kept me, kept me in
Mm.
food until I was able to get another job.
You also know David Niven and his sidekick,
Oh, you mean Larry Niven and Jerry
Larry
Pournelle?
Niven,
Sure.
and Pornell. Yeah, yeah. David, what's the first name of Pornell?
Jerry Pournelle and Larry Niven.
Yeah, and you also knew H.P. Lovecraft, I understand, right?
Well, it's funny you would say that because I have one of my own. There you go. Right.
So Mark made this for me, hearing that I was a Lovecraft fan. He 3D printed HP. So I have a lot of 3D printed things from Mark.
Oh, yeah.
Here's another. Oh, you have a phage, right? This is Thulu,
Right.
right? This is Thulu.
That's Cthulhu. Yes.
Thulu.
Or as I'm told to say it, Cthulhu.
Thulu, yeah, well, we can't pronounce. And then this is your favorite organism, the tardigrade, right?
What you mean, like this?
Yeah.
Yeah.
And you have a tardigrade tattoo, don't you?
I do, but I'm really happy
Oh, look
with
at
this.
that. A Mark Martin tardigrade. That's great.
We call them tardigrades.
Oh, that's
Yeah.
wonderful. And so Mark is a microbiologist. He loves microbes. In fact, his shirt says microbes are greater than humans. Look at that. Microbes are greater than you. Mu greater than you.
And if you
This
look closely, the mu is made up of microbes, the u of people, and it's power, prevalence, and gosh, I can't see the first one. It's wrong angle for me. How about that? You don't
our
often see
prevalence.
that. There it is. Anyway, that's me.
The you get your classes to make interesting things, right? Like
Yeah.
badges, buttons,
Mhm.
right?
Well, you have a lot of my buttons. I really think that promoting microbial literacy is important. And so one way to do that is I make stickers and I make buttons. And I've worked with a lot of really remarkable artists who've been able to create things for my students. And so we make T-shirts for my micro students every year, that kind of stuff.
I'm wearing a spike shirt tonight.
There you go.
I bought it to match the glasses, right? You said you like these glasses, so the T-shirt, the spike shirt matches the glasses. And I have to remind everyone that Mark sent me this recently. Count Phagula. We named it last time. It's very cute. It's very
Yeah,
fuzzy.
there you go.
Did you know this is by Kyrgyzakt,
Hmm.
a very interesting science channel on YouTube.
It is, and I didn't even know that they made those, Vincent. And I was just like scouting around and found them.
Yeah, well, everybody gets into the swag act, right?
Yes.
Because make money on it. What the heck?
Well, I
But
don't, but yes.
so Mark
Yeah,
has a laboratory, and he quality quorum,
that's
and
us.
he studies a variety of microbes, and he's also interested in bacteriophages, viruses that infect bacteria, because they do infect the hosts that he like. And Mark is starting his own podcast, and so I thought this would be a good time to bring him on. In fact, if you notice the thumbnail when we started, that image on the right, a quality quorum podcast. You're going to call it Matters Microbial, Mark? Is that the idea?
That's right.
And
Isn't the artwork lovely?
the artwork is lovely. Who did that artwork, by the way?
That's Liza Vanda Art. And I love the name. And Liza is a really wonderful, wonderful person. And she's done a fair amount of art for me. And she did that just on the anticipation of me doing this one of these days.
Well, it's going to happen because you said you gave me the go-ahead to produce it for you. So it's
That's
going to
right.
be a Microbe TV podcast. And what's the format? What are you thinking about?
So the whole thing is supposed to be relatively short, like 20 minutes or so. And I'd like to introduce one type of concept in microbiology. For
Hmm.
example, something like what is a colony or what is gas phenomena, that kind of thing. And then the other 10 minutes or so will be an interview with somebody. I've got well over 20 former students of mine who are doing great science, and they're all ready to be tapped for that. I actually know somebody I'm sitting next to here virtually who's going to be my first guest. That would be you, Vincent.
I'm not
And
your
we'll
former student, though.
Well, okay, that's true. But then again, you know what I call my former students?
Mark adjacent.
No, they're Doc Martians.
Ah, that's good. I like that. That's very good. Yes, I would be pleased to be your first guest, and hopefully we can do it next week.
That's what you wanted me to do, and that's what we'll do.
And then we'll get the ball rolling. I have a producer for you, an intern for the summer, and that's Julia, and she will get it rolling. And then it's up to you to. We made a schedule, if you might see on a Google Doc, so you can schedule it and put it there, and we'll know when you're going to do it. You just need to account for the 3-hour difference.
That's right. And I'm looking forward to it because there's lots of very interesting things to talk about. And, it's my chance to chat with people who, some of whom might be microbiologists, some of whom aren't, about some of these issues. Because there's a lot of, I don't like to call it misinformation, but it kind of is out there. And I like to talk about that.
So, folks, queue up your microbial questions for Mark. He can
Yeah.
answer them for you. So, one of your interests is predation, right?
Absolutely.
Who's predating who?
Yeah, it's an interesting question. And there was a scientist in the 1960s named Mortimer Starr in the early days of Bdellovibrio work, which is one type of bacterial predator. And he wrote a 20-page paper on whether we should call Bdellovibrio a parasite, a predator. You had all these different things, a parasitoid. And you realize after you read it that you lost an hour and a half of your life. And it really isn't important one way or the other. Fact is, nature is all about getting energy and resources in one way or another. So it's not a surprise that there are all kinds of predators out there. I've talked about them on TWIM with you several times, Vincent. And turns out there are ones that go after the cyanobacteria in some of the rock varnish. I sent you a link to that paper, which is fabulous.
Yep.
So everywhere you look, you find some bacteria that take advantage of others and try to live off of them. And some people are very interested in trying to develop these predators into being living antibiotics. And so there's a lot of hope for that.
Yeah, when you come on TWIM, you call it being TWIM adjacent, right?
Yeah. Well, I'm not part of the twimumvirate. So there it is. I can be adjacent.
Well, you should come on and do that paper that you sent me. That's a good one.
Oh, Anna's making me blush. Oh, Anne, you're so nice.
Doc Martin is one of the most wonderful educators I've ever known. He makes science accessible and fun. He inspires me as a science teacher to this day. That's what you need to do. You have to inspire people. And folks, you can ask virus questions for sure. We will Mark doesn't mind if we answer virus questions. And here's one from Kang. This is a more broad question. Could selective breeding of crops and farm animals be considered gain-of-function experiments? Is evolution a gain-of-function experiment of nature? And Mark is nodding yes, because that's the answer to both, right?
Yeah. Vincent, are you familiar with bithorax in fruit flies?
Yeah, I know the gene, yes.
So that's a single mutation that allows a dipteran, a fly with 2 wings, to have 4 wings.
Right.
It's what's called a posterior shift of fate mutation in developmental genetics. And what's really cool about that, if you look in the fossil record, you see this sudden shift from things with 4 wings to 2. It can happen. So you want to call that
2 wings to 4?
Yeah.
And so because that's better for flying, right?
Well, I think it depends on the environment. I mean, beetles have 4 wings, and everyone knows that nature loves beetles.
Nature adores viruses.
That's the old, the JBS Haldane quote about that I like so much is they asked him in the 1920s, he was a famous atheist, and someone at the London Times asked him what, if anything, his research in genetics had taught him about the mind of the creator. And he said that God has an inordinate fondness for beetles.
That's right.
And that's because there's so many of them. But if that's showing a fondness for beetles, what on earth do you think with all the microbes? And if there are more viruses than there are bacteria, certainly. So there you go.
Well,
Vincent, did you know that I did my PhD work on bacteriophages?
I don't think I remember, but you probably told me. Yeah.
No, I made a generalized transductional system for Cynorhizobium meliloti in grad school,
Mm.
which is still used to this day for moving genes around. So I had to go find phages in nature. So they're lots of fun.
Sometimes I title my talk, depending on the nature of a talk that I give, an inordinate fondness for viruses. But I'm
Yeah,
not referring to the creator. I'm referring to me,
to you.
because I do have an inordinate fondness for viruses. Anyway, Kang, yes, selective breeding, if they have a new property of
Mhm.
any sort, and that's what selective breeding is about, there'll be gain of function. And evolution, a new property is a gain of function for sure. And that's
I
why the discourse around these days that gain of function is, oh my God, they were doing gain of function as if it's only a bad thing. I never met a gain of function experiment I didn't like.
So
So,
look at teosinte, the ancestor of corn, maize,
hmm,
and those are certainly gains in function.
sure. No, it happens all the time. So, yes, that's a good question. And I'm glad you asked. But unfortunately, only the people here are going to hear the answer. And not the rest of the world, which really needs to hear it for sure. Okay, let's see if there are any other. There's a lot of chatter because people love to chatter. So Jessica says, "Greetings, Earthlings. Couldn't help myself after watching 1979 Alien, which it took like 20 minutes to see the first human or something like that, right? You see the spaceship and then machines and this and that. It's a really amazing movie, considering." Okay, let's do a virus question. A recent New England Journal study did biopsy on taste papillae and used in situ hybridization for RNA as evidence for the presence of virus in cells. Is this RNA test different from PCR for virus versus debris? That's a good question. It says there's RNA there. It doesn't say there's infectious replicating virus. It's the same as PCR. It tells you, RT-PCR tells you there's RNA there, but it doesn't tell you that it's from infectious replicating virus. So, yeah, there could be virus there. Now, there is a little twist when you do in situ hybridization. You can look, let's say you're looking at for SARS-CoV-2 RNA, you could look for the negative strand instead of the plus strand, which is in the virus particle. And if you look for the negative strand, that means that at one point there was some replication going on. It may not still be going on, but it's a little better than PCR in that sense. Does it bring back your phage days, Mark?
Oh, absolutely. Um, I have so many cool stories about that. I don't know if folks are interested, and I don't know.
Tell a cool phage story. They love it.
Well, so when I was first working with this bacteriophage, now this is in the early days and I didn't have access to ways of looking at very large pieces of DNA, and I wanted to know the size of this bacteriophage genome. And so I thought, well, I'll just cut it with enzymes. And the first thing that I found with restriction enzymes is that there were some frequent cutters, that is to say, enzymes that cut at 4 base pair sequences where I got really huge pieces of DNA. And that's because the way that restriction modification systems work. So the phage is methylating its DNA, and the defensive enzymes, because that's where restriction enzymes are from, they're like a primitive immune system for bacteria. And so they were actually not able to recognize those sequences in the bacteriophage when it injected its DNA.
Mm-hmm.
And then I cloned a piece of that DNA, passaged it in E. coli, and found out they still didn't cut because that just so happens these 4 base pair sequences were very rare.
Yeah.
And it just made me think how clever, and I don't mean that in terms of thinking microbes, but how clever nature is with this kind of business. And that's when I first started saying Darwinowing, by the way, was then. How
Darwinowing.
evolution has kind of filed away at it.
It's interesting, you call it a primitive defense system, but to the bacteria, they would be offended. They don't think it's primitive. It's pretty good stuff, right?
Mm-hmm. Most bacteria have several restriction modification systems, and we haven't even talked about CRISPR. So the fact is, they have to defend themselves from invading DNA from the environment.
Yeah,
They
yeah.
have to tell their sibs from non-relatives, kind of like we do during organ transplants.
There are so many interesting defense and counter defense systems in the bacterial world. So Andrew wants an image of my room as a Zoom background. Okay, I will take you one later, without me, of course. That'll be easy. I'll just hit the timer and get out of the picture, and I'll send it to you, and you can use it. No charge. No problem whatsoever.
Oh, boy.
Hi, Mark from the University of Toledo in Toledo, Ohio.
It's common that I've run into people who've met me, and I don't know them by only their name, but need their face as well. So, Jessica, if I've met you and don't remember, my apologies.
I know I sometimes have the same thing because a lot of people hear us and we don't necessarily know who everyone is. But here, it's usually the same people every week, and I've gotten to know most of them. Now and then there's a new person, but not always. You can also tell stories about famous scientists, can't you?
Yes. What would you like to know?
An interesting one. You told me so many. I had dinner with Mark in Houston a few weeks ago. He told me so many interesting stories.
Well,
It's great.
I mean, I got a chance to meet people as there's a saying in physics that physics advances on gravestones. That is to say, as famous people in the past move away, then
Mm.
there's space for new people. So I was fortunate to be at Stanford in the 1980s and got a chance to meet some really remarkable people like Alan Campbell, who was on my PhD committee, Charlie Yanofsky, who was not on my PhD committee. I was petrified of him, who discovered, well, first off, Alan figured out that the episome model of how phage lambda and other types of prophages can sometimes form. And Charlie Yanofsky and his group were the ones who figured out attenuation of amino acid biosynthesis, the first time you could think of an RNA molecule acting as a regulator all on its own. It was really remarkable. I had a chance to meet any number of people. Esther Letterberg, who I had lunch with her many times, and we talked about the early days with Joshua Letterberg, determining the nature of mutation, the discovery of phage lambda. Was remarkable. I also have stories that are spill the tea type, but those I don't generally broadcast.
Yeah, I can understand that. You don't want to get people mad.
Oh.
We
But
invited.
scientists really are people, and there are good scientists and not so good scientists, and just like any other job. And it's important that everyone remember that.
We invited Vincent and Amy to Bologna. I do remember, but I haven't gone there yet. Ron is from the Bronx originally. Very good. Love to hear it. He from the hood. Yep, he's from the hood.
Yes, the flow hood in the lab, right?
So do you here, Doreen says it's great to be a writer and to know writers.
It was interesting. Yeah.
Do you still
And
keep in
it's
touch?
it's fun. It's kind of a thrill the first time you've worked with a writer as one of their readers and you've discussed various ideas and they put you in the acknowledgments, and it's such a thrill when that happens. It doesn't really matter. It just makes you feel funny to see it.
But
Um,
you don't write any longer, right? Fiction.
I don't have any time when I'm teaching to do that, and like the publishing industry has fashions and lack of fashions. And here's a surprise: I'm an unusual person. I know none of you knew this, and it doesn't really fit with some people. I've had this experience many times in my life, but if I were one of those writers that no one's ever met,
Hmm.
maybe I could get away with it.
Do you have any of your cool badges in front of you that you could show folks?
Oh, let's see what I've got handy here.
What's the thing on what does the thing on your lapel say? Being yourself
Oh,
is an asset, right?
it's the best. Um, oh, I got my angles all messed up, so I'm just going to do it this way.
Being yourself is best. Yeah, that's
And
good.
it glows in the dark because everything should. And then I saw this on Etsy, and isn't it beautiful?
It's got
It's
microbes, doesn't it? Yeah,
it's
it's nice.
pretty and it's beautiful.
Very nice.
Yeah, yeah. I have quite a few of those.
Tom says, laid off from biotech. I know that dance.
Yeah.
It's a slow dance, isn't it, Mark?
It is a slow dance, and but it's okay. I mean, there's biotech and biotech. I worked for a company in San Diego called at the time Kelco, and what
Mm-hmm.
they did is they made xanthan gum, which is a polysaccharide from a bacterium, Xanthomonas campestris. And one of my jobs, by the way, Vincent, was to make the bacterium phage resistant because every once in a while they would get a phage infection in those giant fermenters and everything would turn to froth.
Yes.
And I told them if you just wouldn't put the exhaust in the sewer, this wouldn't happen. But they never listened to me about that. And they didn't understand
That's
that
great.
they thought there could be one phage resistant Xanthomonas that would be resistant to them all. And I said, it's not like that.
Mm-hmm.
Yeah.
Patricia said, I saw this on Etsy. My fave comment so far. You just said that, you know.
Yeah.
Professor Mark at UCSD, did you ever cross paths with Dr. Seti or Dr. Crotty across the street?
Neither.
It was before their time. I think you were there a long time before they got there. I mean, that's the La Jolla Institute for Immunology. I don't think it existed when you were there, right?
No. I did go to Scripps Clinic a fair amount, and I went to the Salk Institute quite a bit. It's lovely. I couldn't afford to live there, but there you go.
Steve in England says, does Mark watch Nick Zenter's live Washington geology YouTubes? They show the world your wonderful state scenery. Vincent and Nick are two of a kind in teaching the whole world. Oh, thank you so much. Have you seen this channel,
I have
Mark?
not, but I really do love the geology of this state. It's really interesting. And it's funny that I ended up in one of the few places there aren't a lot of fossils. There are only a few, because I love fossils. And there's only, no, I've got a couple of them over here, don't I? Oh, I have meteorites, and oh, I've got all kinds of things here. Hey, wait a minute, Vincent. You haven't seen this. Hold on. This is just for you, Vincent. What you're looking at right there
Hmm.
is a fragment of a Martian meteorite.
Wow.
This came from Mars.
So the dark part is the meteorite.
Yeah. And then this is the one type of meteorite that I don't have in my collection. This is a small fragment of a carbonaceous chondrite. So these are the meteorites that have organic material in them.
Cool.
I don't know why I'm telling you about that when I was meant to talk about fossils, but I have a lot of bric-a-brac. What's the old expression, Wunderkammer, that the old Germans used to have, this cabinet of curiosities? It's a really cool thing.
One of my students from the spring went on a trip and she brought me back a piece of the Grand Canyon and a piece of something else from the desert. I don't remember what. Very interesting rock formations. One of them has 3 layers. Very cool. So Doreen feels excited but a little out of the loop with tonight's vibe. No, Mark is a wonderful microbiologist. I wanted you guys to meet him because he's a funny guy. He knows a lot of stuff. He's not a virologist. He doesn't do Racanielling, do you, Mark?
No, I never Racaniello.
By the way,
I
Mark
didn't.
thinks I should do 15-minute podcast every week called Racanielling,
Yeah.
where I just rant about something, right?
I think it would be very therapeutic for you.
You think I need therapy, eh?
No, I just said it'd be therapeutic.
It would be.
I didn't invent Racanielling, though. Who did?
Someone wrote it in during the early days of the pandemic when I was complaining about something. We love to hear Racanielling, and it stuck. I think it's a cool thing. But I calmed down since then. I tend not to do it, but certainly things get me riled up. I could do that. So we're going to try it. I ran it by Jerry, who's so I had a lady in my virology course this spring who is an established science journalist, and she's going to work with us. And she likes the idea, Mark.
I think it's great. And can I tell everybody? I mean, I've had several people ask me how I met Vincent, and I just walked up and we started talking.
It was at an ASM meeting, right?
Yeah.
But you'd probably, I'd heard of you because you're out there with social media, and you'd heard of me, so it was just a natural thing. So you think,
Yeah.
when we go to ASM meetings, American Society for Microbiology, Mark is always surrounded by people, and he's handing out stuff.
Yeah.
Mostly buttons, right?
Yeah, I actually spend when I say a fair amount of money, it's not like I'm destitute because of it, but I print out like OMG for overwhelming microbial greatness, or WTM for what the microbe, and there's new buttons every year, and again made by wonderful artists, and I just hand them out to people because Vincent won't believe this, but I'm a little shy in groups, and it helps.
I don't believe it for a minute.
People are really nice, and I really enjoy it, you know?
Well, you have a good vibe, so it's going to make people comfortable. Yeah.
Yeah. So, I mean, but I love microbiology, and I always have. And so it's really a wonderful experience to go to these meetings and see very famous people. I mean, Elio Schechter was my hero. Is just the greatest. I used to love it when he would get upset about something and curse, because when I curse, it doesn't sound elegant. And when Elio, it would have that beautiful, almost continental vibe to it. I was in awe.
Yes, it's got a good accent. Very good.
Yeah.
Wandering World says, I'm beginning to think I'm the only person without a phage knocking about in my room. Well, you need to get one.
And if you're careful, you can get one that flashes lights on the inside. We call them disco phages.
Yeah,
I want to get.
I have
There
one
it is.
from Mark. Let's turn it on.
One
Turn it
of these, the batteries won't work.
Let me turn off the comment. I bet it works. Yeah, there
There
you
it
go.
is.
See, it's cool. Actually, some of the light thingy is coming out here, Mark.
Yeah. Just shove it in with a pencil.
It's almost like intestines coming out. That's cool. And I think if you push it again, it goes steady. No, slower. And then if you
And then
push it
one more
again,
time,
it's
right?
on all the time. Let's turn the light off and see what this looks like. Are you ready?
Here it is. Look at that, folks.
Holy, let's put it on flashing. That's a little too fast. That's a little nerve-wracking. There you go. That's better. And then we have our on all the time. Very cool, Mark. Thank you so much for that.
So what I like to do is my wife is really quite skilled at 3D printing, and so she was able to modify one of these so it's hollow on the inside, and I would just stick those lights up inside.
Mm-hmm.
And my dream one day is to have them be like sound activated, so they will go faster or slower depending on sounds around. That's a disco phage.
Actually, Tom says you need it to be multicolored.
And I have done that. The supplies in China of those tiny little lights are not as good as they used to be.
I'm sure that you could get anything you wanted. Yeah. Okay, here's Carol, our favorite nephrologist. She says, beyond the noise, chemophobia. If Del Bigtree is worried about the 25 milligrams of sodium phosphate in vaccines, he needs to know that phosphate additives contribute 300 to 1,000 mgs of total daily phos intake. Well, Carol, he probably knows, but he doesn't want to know. He doesn't want us to know. He just wants to scare people, as
Yeah.
Paul Offit said. Yeah, we take in a lot of stuff.
I mean, do you remember there was this scare about aspartame?
I do.
And what people would say is it would make formaldehyde because it's just two amino acids linked together. And in the process of cleaving them, you can release formaldehyde. And that sounds terrible. It turns out that the amount of formaldehyde in orange juice is hundreds of times higher than you would get through the metabolism of
Mm.
aspartame. I'm not saying that you should like an artificial sweetener. That's not what I'm saying. What I am saying is there's no need to be scary about it.
But people will scare you nevertheless. That's the modus of the world these days, to scare you.
Mm-hmm.
Marty says, scientists never grow up. Amazing. They still love the toys.
Why not?
Well, I have to admit, I didn't have many toys until Mark started sending to me. Right?
So it's my fault.
He seems to understand that I have a need for these things, because otherwise I wouldn't have very much. No, I'm looking at my shelf here. It has a lot of Mark Martin toys on it.
Yeah. I mean, people send me stuff, which is kind of fun. Well, they just like I sent you stuff. Like, Vincent, have you seen the tardigrade that somebody sent me? Like, you've heard of a 6-pack? This tardigrade has like a 12-pack or something going on.
That's good. Is that a 3D tardigrade or something
Oh
else?
no, this is needle felted. It's beautiful.
Oh, okay.
Yeah, I
Very
can't get
nice.
the number there. Oh yeah, and I'm not paying any attention to the questions that are coming along the sides because I thought Vincent would put them up. I see so many cool things going by. So those of you who are there putting in questions, I love them.
I'm selecting them, and I'll get to many of the ones that you're out of focus. There you go. Now you're snapped in. All right. So Les loves the one topic short podcast. So you'll get that from Mark, right?
You will.
I told Julia there's no way Mark is going to keep it to 20 minutes. So we'll see. She laughed.
Wait till I start giving Julia all kinds of microbial swag. Then she'll be on Team Mark, I promise.
We have a virus question from Peak. Do you like his name? Peak Dunning-Kruger?
Yes, I do.
It's not his real name. How does the damage caused by parvovirus entry affect the cell? Are there cellular repair pathways for the nuclear membrane, or is the damage dealt by the parvo irreversible? So what Peak is referring to is that the little parvoviruses, they bind at the nuclear pore, and then they cause disruption of the nuclear membrane in order to get into the nucleus, where they have to go to be reproduced. And unfortunately, there are no repair pathways for that, as far as I know, and that is bad, because then things start to leak out of the nucleus, and eventually the cell will die. R.I.P. cell. Yes, Mark.
I thought it's interesting because during mitosis and meiosis, depending on the cell type, the nuclear membrane has to go away and then come back. So you would think they would have mechanisms for fixing damage.
They make a new membrane, as far as I know, right? Just make a new one.
Yeah.
I don't know.
So
I'm not aware that they can deal with holes.
Yeah.
But it's a very good point, though. Yes. John wants to know, do you recall the first piece of microbial art that caught your eye?
Well, do you mean microbes by themselves, or do you mean a piece of art that a human made?
Well, let's start with the human-made art.
I believe it was a painting that I saw of Louis Pasteur with one of those gooseneck flasks that I would love to have a replica of.
Oh, that's cool.
Yeah, but when I was a little boy, I remember looking through a microscope at Spirogyra, which is a beautiful alga that has like this helical chloroplast running through it, and it's slowly rolling. Same thing with Volvox. And they're large enough that you don't need a fancy microscope to see them, and it just took my breath away when I was like 4 or 5. And since I also love astronomy, The stars in the sky, the cells on the plate. I mean, this is like universe to me.
Spirogyra, wasn't that a band?
It was smooth jazz, Vincent. Your jam.
You know your music, don't you, Mark?
I know a little bit about a lot of stuff.
Oh boy, yes. I've had that told to me before. A lawyer once said, "You PhDs know an awful lot about a very little, don't you?" Kind of insulting, right?
People are, and they'll say, "Don't take it personally." That's my favorite. My favorite
How
response.
else?
And then I say, as soon as you're not personal, I'm happy to.
How else can you take it? Luis wants to know how to find your podcast.
Well, it's going to be under your umbrella, correct?
It is. It's going to be at microbe.tv.
Mm-hmm.
We'll make
It's
a page for Mark. And what's it called?
Matters Microbial.
Maybe I'm going to make a little thing here. Matters. I'm going to make a little text that we're going to put here. So it's going to be on the Microbe TV site. And he's recording the first one next week. So you should see it before the end of the week.
Yeah.
With me as the first host, unless he changes his mind. He could.
Oh, no. Vincent, you're the podfather. Come on.
The podfather. I think that's great. I have to bring up Anne's comment here for the record. One of the most wonderful educators I've ever known. He makes science accessible and fun. He inspires me as a science teacher to this day. So what level does Anne teach of science, Mark?
Middle school.
And Anne was an undergraduate in your laboratory?
That's correct. And then she did a master's degree in education and worked with me a little bit on that. We talked about Winogrodsky columns and things of that nature. It was a wonderful time. Anne is great.
We need more teachers like Anne who have been educated in a good laboratory, right? Mm-hmm. And understand how science works.
Absolutely. So when I taught microbiology at that time, and I still do this a little bit, it's an upper division course, has a small number of students, and then they would get to investigate for like the last third of the course some question that they want to come up with, that they design. It could be, and this is because of the amount of coffee I drink, another surprise, friends. People would try and find microbes that could break down caffeine. And we would talk about enrichment culture and how to do that. Other people would try and see if microbes could survive in soda pop because of the pH issue. And it was a really good way to have them think about some ownership of things. You have to be a little bit careful with that approach because some students want to do too much or too little. But we had a lot of fun in Anne's class with various people there. That was a long time ago, though.
I want to just refer to something Mark said. He called me the, not Godfather, it's the spell checker changed it. Podfather. Technically, someone else has been called it before me, but Mark wants me to be called the Podfather. So thank you very much, Mark. I appreciate that.
You're helping me. It's a wonderful thing. You're giving me an outlet because I actually find it really therapeutic to talk about microbiology with people.
Talking is great. I really like it. Yeah.
As you say, it's the conversation.
It is the conversation. Here's Barb Mack. I only heard of tardigrades recently as I've been watching the Icelandic volcano eruption where lots of moss has been on fire. Could they survive that?
I make a joke about that. What do you call a group of crows? It's a murder of crows. It's a crash of rhinos. And a friend of mine, Katie McKissick, came up with a badass of tardigrades because they are so incredibly extremophilic. I don't believe they could survive being burnt up, but they can survive in their cryptobiotic state when they're kind of in suspended animation, can absolutely survive vacuum of space, and they absolutely can survive deeply cryogenic temperatures. We know that they can for decades. So tardigrades are awesome.
So, Mark, you see the little blue wrench next to her name there, Barb Mack's name, on the screen?
Yes.
So that means it's a moderator.
Hello, Barb.
They get to fix things. We have others, of course. Here, for example, is Steph. Welcome, Steph. Steph joined after I thanked all the mods in the beginning.
Do you
Here's a
like this
blue.
one, Vincent?
That's very nice. You have a lot of tardigrades.
Well, yeah, it's a tardigrade army. This one's articulated, which is kind of cool.
It is. How many do you
Yeah.
have? Like 10?
I imagine I have 40 or 50.
So Pete wants to know if cholera a Vibrio bacterium.
Yes.
He has a pic of the Jon Snow Monument on Broadwick Street, London.
Vibrio cholerae, absolutely.
So that was an outbreak in London, which he stopped by taking the handle off the pump, ostensibly, right?
And that's a really interesting organism, the way that this is something that Elio Schechter has talked about a lot. We've learned a lot about how our bodies work because of the way that bacteria interact with them.
Mm-hmm.
He calls that cellular microbiology, which confuses people, but it's accurate. So we understood a little bit about ion flow because of the effect Vibrio cholera has, because it hijacks the normal way of controlling fluid flow across the gut epithelium. And that's why you get the massive amount of fluid loss, which is really what causes, God, I sound like Michael Schmidt. It really is responsible for lethality, is just fluid loss, which is just amazing to me.
Well, this is an interesting one. I just listened to the latest TWIV, and oddly enough, also just finished Kahneman's Thinking Fast and Slow. You know that book, Mark?
I own it. I have not read it.
Yeah, it's very good. Curious on your thoughts on what the System 1 versus 2 differences on vaccines are. I haven't thought about that. I think the anti-vaxxer response is a System 1 response with no input from System 2. And that's why it doesn't make any sense, because it's just a gut reaction that, nope, I'm not putting that in my body. And you'll see when you read it, Mark, what this is all about. That's a good question, Jeremy. John writes, through this community, someone suggested a good primer book on biochemistry that I'm finding useful. Do you have a go-to recommendation for a primer in microbiology, Mark?
So that's a really, really challenging question, and I'll tell you why. When I earned my bones as a microbiologist, a man named Ed Ledbetter, who was my instructor at the Woods Hole Microbial Diversity course, said that every microbiology instructor needs 3 textbooks, 1 for the student and 2 for them. And it's a hard question because to me, I want to know everything, but not to most students. They want to know a small amount. So I find that the book Microbe that ASM Press puts out is a nice soft cover. It has more information than you probably need, but it's not quite as pricey as the other ones. Like Joan Slajewski does a wonderful book on microbiology, but it gets a little pricey. But no one's, I mean, I think you could probably find like the Dummy's Guide for Microbiology, but who wants to read that? What you want to read are the things that are exciting.
Okay. Oh, look at this is a very interesting comment. Everyone who gets infected is a gain of function experiment.
Wow.
Yeah, you're getting some additional genetic material.
Yeah.
I mean, the key is whether you get a new function or not, right, of some kind.
Right.
But you could argue that at the cellular level, you're getting some new functions. So, yeah, I would have trouble arguing with that. What do you think, Mark? Would you have trouble arguing with it?
I don't. I mean, when there's absolute truth. I mean, have you told them about what happens with the placenta and retroviruses?
Sure, it's in my course. Yeah.
Right. So that's a really good example of an infection at one point that led to a function which pretty much changed developmental biology. But would every single person who gets infected with a virus have that change? Again, the change in the phenotype, what you observe, is important with that. And I'm not sure every individual that's infected will have that. I mean, for example, I came up positive for
Mm.
COVID-19 last year, and I was completely asymptomatic. Which,
So
Vincent, I've had my genome done, and I'm curious if I have those new markers.
yeah, if HLA-B15, yeah. Well, you did not test positive for COVID. You test positive for SARS-CoV-2
Thank you.
because it's hard to
No,
test for a disease.
it's an antigen test, and I think that precision is important. Some people will kind of roll their eyes about stuff like that, and I tell them, in everyday life, we're learning to be cautious about what we say because we want to be precise. How is this any different? I think being precise is
Yeah,
really
I
important.
think you have
And
to
then
use
when
the right
people
words.
say,
Yeah,
they'll say, you know what I mean, but do I? Probably not.
because everyone says a COVID test, right? But it's not a
Right.
COVID test. It's a SARS-CoV-2, but that's too long and people don't want to say it. Here's a good one. Are GMOs gain of function?
Oh, I love that. Yes. I mean, depending on the one you're talking about. For example, 80% of the corn that's made in this country has been genetically modified by agrobacterium to express the Bacillus thuringiensis um, insecticidal protein that we know is really safe and a lot better than spraying pesticides. But that's absolutely something that wasn't there originally. So that's or glowfish. You've probably seen them in aquarium
Yeah,
stores.
sure.
They put GFP or RFP into, originally they were zebrafish, but now they're doing others. That's absolutely true there.
All right, Rob wants to know, what is a philosophical mutation? Did you use that term?
I never have. I love it.
I think it's a good word. I like it.
So
I don't
that's
know
like a
what it means.
paradigm. It's like a paradigm shift, right? So this would be a change in a concept that alters the way that we use that concept. That's really interesting.
Yeah, Rob is Rob, as you can see by his icon, is a fungus lover.
Yeah.
He wants This Week in Fungi.
It's funny how much we ignore that in our classes, isn't it?
I ignore phages in my class. Unfortunately, I mean, we do a few mentions of them here and there, but it is, of course, in animal viruses, right?
I'm just looking on my
Philosoph.
shelf at different kinds of things.
Yeah, yeah,
Like,
no problem.
no, I mean, like, oh,
A book,
this is a
microbiology,
fabulous book, but it's huge,
and
right?
probably expensive, right?
Well, when I say that, it sounds like I'm being critical of the authors, and I'm not. It just happens to be an expensive book. But
Well,
I saw, go ahead.
the pricing is usually the publisher, not the authors.
Right. And I think that's good to say. And I saw, I think it was Anne who mentioned there was a wonderful little book by Bernard Dixon,
Hmm.
Power Unseen, with little snippets of interesting microbes. And there really hasn't been other books like that. I mean, you've got, I think, you've got Ed Yong's Opus and all the rest, which is very good and lots of fun. But I really do like that idea of having a cool collection of interesting microbes.
Ian says, as well as microbes that eat bacteria, are there benign microbes that excrete stuff that are toxic to disease-causing bacteria too?
Absolutely. There are tons of them. We co-evolved with our microbes. So, I mean, a good example is, if you'll forgive me, we're like 3 layers deep of microbes on our skin. And a lot of the microbes on our skin, and they're salt loving because of our sweat, are making short chain fatty acids that actually do inhibit a number of other bacteria that might set up shop. So, I mean, it's not a surprise. Uh, Sure. I mean, that's an example I just gave there. There's lots of them in your gut. I know there are lots of animals and plants that do the same sorts of thing. It's fascinating. There are some people I don't, and Vincent, we haven't talked about this. I don't know where you are with the Seth Bordenstein idea of the hologenome, that we're a collective organism,
Mm-hmm.
not just the animal cells, but everything else.
Yeah. No, I mean, if you look at us, it's our cells, and we're full of bacteria, we're full of viruses, protozoa, fungi, right? We're a holo organism for sure. But we've called coral that for years, right?
Yeah. That's, I think, the first time that was ever used was with coral, because it's
Yeah.
so clear there.
What was
That's
the?
a
Oh
great
yes,
question.
Steph says, Dr. Martin, kindly explain what a microbial centrist means.
Will you forgive the story, Steph and SF? Here's what it is. Um, I was once at a department meeting in my department, and a faculty member, well, group of them, were discussing, if we had more money, what research area would we want to hire in? Now, I want to be clear, I'm at a small liberal arts college. We have small research labs staffed by undergraduates, so it's not the same at as a PhD granting institution. So, in any event, the two people discussing this were plant biologists. And not surprisingly, they felt that perhaps we should have another plant biologist because plants were so important. And I didn't cough. I didn't say a filthy word under my breath. I just drank my coffee, going to my happy place up here. And this one faculty member turns and says, "Well, Mark thinks that bacteria are more important than everything else." And I looked at her and I said, "Oh, I think plants are really important. It's just the really cool things they do were stolen by, from bacteria and archaea." And I'm not wrong, they were. And she looked at me and she said, "You're a microbial supremacist."
Right.
And I said, "Yes, I am." And then that word, supremacy, got very unpleasant.
Mm.
And that's fine. So I kind of evolved that into microbial centrist, because it's a microbial planet. From the as deep down as you care to dig, and as high up in the atmosphere as you care to go.
Yeah, I remember when you were doing the supremacy, and then people didn't like that. Yeah.
Yeah, I would hear about it, and I never wanted to argue with anybody about it. You know what I mean. So I
Mm-hmm.
decided to be more precise.
The theme seems to be blue. I don't think you have blue. You have turquoise, don't you?
I guess I should have checked on wardrobe, right?
No, we
I should
didn't consult before. I just
have. Yeah.
coordinated with my glasses, which are,
Mm.
you know, I just wear these specially for the live stream. I don't know why I started doing it, and then it stuck because people seem to like it. So there you go. Oh, this is an interesting idea. Wandering World says, I think I'm going to start calling it garbled info rather than
Oh,
misinformation, as they often take a paper they found and garble it up until it fits with the hits.
yeah.
Yep. They want it to fit to their idea of how it is. All right. Go ahead, Mark.
That's
Go ahead.
No, I just said that's a really good way of looking at it. I think
Garbled.
if you and it's really hard sometimes not to be really angry at some of the things that people say, but if you want to change minds, we have to do it via positivity as
Mm,
well as fact.
yep.
My opinion.
Um, Silvio Pina says, can Mark speak to the impacts of kids' diminished exposure to pathogens to the increase of allergies? You discussed that on Immune 2. How do you know how much playing in the dirt is too much?
I have to laugh about this so much because Jack Gilbert, currently at UC San Diego, he's a real big name in microbiome work, wrote a book called Dirt is Good. And it's about this topic, which is alternately called the hygiene hypothesis and also called the old friends hypothesis. The idea being that when we were originally around all kinds of potential pathogens, I mean, there are people who don't even like that term anymore, and there's reason for that. But the idea was our immune systems were being trained by the environment. And this is why you find, surprisingly, fewer allergies among people raised around dogs. which is really an interesting idea to me.
Mm-hmm.
So this idea that our immune system is educated by the environment doesn't surprise me at all. Your immune system can make antibodies specific for things that were created in the lab that don't occur in nature. So it's one of the hardest, most difficult topics, in my opinion, in the world. Ecology, immunology, neuroscience, these are the really hard questions. So this idea of playing in the dirt, I think, is important
Is it
because, go ahead.
is it dirt or soil, Mark?
Soil.
Joe Handelsman would insist you call it soil, right?
And I know why, and with very good reason. Again, precision is important because it's the lifeblood of agriculture is to have those things in the soil, and it's so complex and wonderful in there. It really is. One of the things I love doing with students is there are a lot of bacteria. Many of them make antibiotics called the actinobacteria, and they also make a volatile compound called geosmin, which smells just like potting soil.
Right. Right.
And so I'll streak some on a plate, and I'll have them smell the plate, and they'll say that smells like dirt. I'll say not at all. Dirt smells like streptomyces.
So the other story I like is that, which relates to the microbiome, is that if you're delivered vaginally at birth, you acquire the vaginal microbiome and you have no problem digesting mother's milk. If you're delivered by C-section, you don't get that other microbiome. You get skin microbiome, which isn't good, and you have problems digesting milk.
Yep.
It's wonderful. It's just wonderful. And on Immune this week, we did a episode about food allergies and avoidance. And, allergies are increasing. And part of the idea is that kids are not getting their dirt inoculation anymore,
Yeah.
right? We're keeping them too clean. Let them play in the dirt.
Yeah, that doesn't mean if you get a cut to rub soil into it, but it does mean that there are people who don't walk around outside, or they don't roughhouse with a dog or pet a cat.
Yep. I figure in New York City, everyone's getting a good microbiome, right?
So have you seen? You remember the work on that, where the aerosols of what they were getting in throughout the year on the streets of New York?
Yeah.
It was cyanobacteria mostly during the spring and summer, and it was mostly fecal stuff in the winter.
They did a survey of ATM machines throughout Manhattan, and they could find various microbes, but they could also see what kind of food was on the buttons, right? Because people eat and then they have dirty hands and then they go get money, and the food is neighborhood specific. So, downtown where the bankers are, there's a lot of steak.
There you go.
And elsewhere, there's chicken.
Hey, Vincent, I saw somebody. That's the wrong direction.
No, you got it. To your right.
Anyway, it was interesting. Someone mentioned water bottles because I do this business with water bottles in my students, right, where we swab their water bottles, we isolate the microbes, we identify what they are by 16S ribosomal RNA, like a barcode, and people flip out, even though most of those microbes are from them. And that's the interesting part about it. And I call them water bottle buddies, and we made buttons and stickers and things like that. But I've found quite a few, Vincent, that are making antimicrobial agents.
Not surprised. Surprised.
Because they like to adhere in a biofilm to the sides of those water bottles. I also love students telling me how carefully they clean. It doesn't matter. Anyway, there's going to be a lot of competition for that particular space.
Oh, look, Anne was helping John find a microbiology book. Are you looking for an academic or a general interest in diversity? Very good. You trained Anne very well.
Anne is wonderful. And not just because she has a cute corgi either.
Ah, that's a corgi. Okay.
Mm-hmm.
All right, Jan says, how can you distinguish the minus RNA from the plus? Well, it's all in the probe. When you do in situ hybridization, you make what we call a probe, which is jargon, but that's what we call it. It's a piece of RNA that has a detectable thing in it so that you could see it in some way when you look at the tissue sample. It's typically radioactive and you see grains. So the probe, you could make plus polarity, and then it would only detect minus RNA in the cell. And if you made a probe of minus, you would only detect plus. That's how you do it. Nucleic acid is amazing. It's just amazing that, one strand will hybridize with the other only, not with itself. It's very cool. These are the things we take for granted, Mark, but...
Oh, it's beautiful. And then you ask, why are there two different types of viruses? Why aren't they just one? Why, double-stranded RNA viruses, that makes sense. But the minus strand viruses and the plus strand ones, where the plus strand can act like a messenger RNA, the minus one can't. Oh my goodness. All the different things that are necessary.
I know. Well, this is a good question that students often ask. What is the reason for so many different genomes? And I tell them, well, if I were the creator, I would have them all plus-stranded RNA. It's the most simple and straightforward and elegant. The RNA gets into the cell, it's translated, and you make more viruses. You don't have to go in the nucleus. You don't have to make the other strand. It's very simple, but it's not the way it is. Everything works. Whatever works is out there. There's a niche for it. On a planet as big as Earth.
This reminds me of the joke that I told you recently, Vincent, where a student would ask me about how viruses evolved in the 3 different models and which one I thought was correct. And I said, yes.
Yes. Yes. Good answer. Yep. Yes. Which is correct. Yes. I like that. So Andrew says, I still play in the dirt. I just call it gardening. Yes.
Absolutely. I mean, and depending on what you mean by your soil that you're playing in, I mean, there's like dry, dusty stuff, but that wonderful gardening soil, that's a lot of fun.
So, Mark, is there a role for continual encounter with dirt, even if you're older, for microbiomes?
I imagine there is. It would be, I know as one grows older, the immune functions in general tend to lessen.
Mm-hmm.
And I'm wondering if you had people that were constantly into gardening, if that process is retarded in any way. And that would be interesting to me. And I wonder if there's data like that out there.
Yeah. No, I agree. We do senesce. We do immune senescence. You and I are immune senescing, Mark. I hate to tell you.
And I'm well aware.
And it could be that, well, no amount of dirt is going to restore our bone marrow, but it could help with other things. Yeah. Here's the book that Anne recommended for John, Power Unseen, How Microbes Rule the World. You use that book in your course, Mark?
Oh, not for many years. It's out of print now, and it was written, I think, 25 or 30 years ago. It has these very short, punchy, like page and a half long profiles of different kinds of microbes.
Yeah.
So, for example, Halobacteria quadratum, which is square, magnetotactic bacteria, that kind of thing. It's a really interesting book, and I used it for quite a while. It reminds me very much of the way that Carl Zimmer would write about parasites, where he had lots of different parasites he wrote about. And again, I think Ed Yong tried to touch on that. It's not just broken up into little sections. That's the only difference.
I like March of the Microbes.
Yeah.
By, who's the author?
Oh my goodness, this is gonna. I mean, he's a professor at UC Davis. He's one of my favorite people.
Yeah, I'm gonna look it up.
Yeah, because he's awesome. March of the Microbes was great.
John Ingraham.
He's the best.
Anyway, I like that book. Each chapter is a different environment on Earth, and he explains the microbiology of it, and I really like it. And that is asking for a 20-part documentary to go to each one of those environments and record a presentation and talk to experts, and that would be so cool.
Wouldn't it be like be Richard Attenborough going to different environments like that? And they tried to do that with an old series that ASM put out, going to different environments for things. But I think there's time for young.
I understand that his accent and demeanor would be good. But I want to do it because I'm enthusiastic.
Well, let me tell you something about John Ingraham that's a good story because I love it. In the 1960s, in the early days of Bdellovibrio work, because he did some work with Bdellovibrio, John did, and I love this story and I confirmed it with him. There is a very famous person from Germany who I won't name, who was very formal and came to work in this lab at UC Davis. And there happened to be a woman who was a graduate student there, and she had a tendency to whistle while she worked. And this very formal German gentleman did not care for this, and he complained to John that his student was being disruptive. And John thanked him very much. This is in the middle of the 1960s, friends. And the next day, when that very formal Germanic personage arrived in lab, everybody whistled because John had talked to everybody.
That's great.
And
I like that.
That's why I adore John. That was the best.
Artemis wants to know why we need a different vaccine for shingles than for chickenpox. Well, we don't, but that's the way they were developed. The chickenpox vaccine was developed first, quite a few years ago. It's an attenuated infectious virus vaccine and given to kids. It's not for a while. It was used in a higher dose for a shingles vaccine, but it didn't work very well. And that's why Shingrix was developed as a shingles vaccine, which just gives really, really good protection against disease. And that's why we have two. And in fact, now Shingrix is being trialed as a chickenpox vaccine for kids because it's so good. And we may get rid of the infectious attenuated virus. We'll see after the clinical study. So sometimes the way things are developed just determines what's going on. It's a good question.
And I got Shingrix.
Did you get twice? I guess you would, right?
Yep.
Would be nice to hear a discussion on viral neutral selection theory in the face of intrinsic cellular defense as virus spills over or cell tropin happens. Well, that would be a whole podcast on its own.
Well, it's a great question. It reminds me very much of the idea that people have that when a pathogen, and I'm using that term, I apologize to Arturo Casadevall for using the word pathogen, but you know what I mean by that. See what I did there? Anyway, when it encounters an organism for the first time, it's very serious, and then it evolves toward being less dangerous. This is not true, and it's something that everybody is convinced is true. And it kind of suggests there's a directionality to things that doesn't really exist. So this idea of how organisms become resistant is an interesting one. I have a lot of fun with. I think I've told you, Vincent, the phage lambda work that we're doing, where we're trying to see if we can identify specifically where the mutations take place in that receptor gene in a laboratory setting. As a student outreach project is kind of, the issue with the deletion in the CCR5 gene involved with HIV. And this is something that the students might be able to get a good handle on in a laboratory setting. But there's always
so you're talking about the deletion of CCR5. And what's really interesting about that is we don't know what selected for it initially because it's been around much longer than HIV has. And we don't know. We just don't know.
I mean, what a lot of people have looked for the presence of that allele in different parts of Europe.
Yeah.
And I think what a lot of people believe is something that was immunologically similar to HIV, went through Northern Europe about 1,000 years ago, but who knows?
I know that people thought it might be plague for a while, but it's not it.
No.
So we don't know. Elliot says, so we have AI Armageddon looming on the horizon. Where does antibiotic resistance come in and how might its trajectory be altered by AI?
Yeah. Well, a lot of people have started using machine learning to look for new antibiotics. This is a huge issue because it's not that we don't know how to find new antibiotics, is that and I'm not trying to be political here, but most of the companies aren't interested in developing them because we're a very litigious society. there needs to be constant new antibiotics because the antibiotics don't change. So organisms can have a mutation that prevents the efficiency of that antibiotic effect. And that's why we have to keep looking. That's why the folks at Tiny Earth that Joe Handelsman has championed for a long time are using students to look for microbes in various places that have antibiotic activity, with the idea that there'll now be lots and lots of new ones to look at. But I also know that people are starting to use machine learning and AI to see if they can develop completely synthetic drugs that might be able to work properly on some of the pathogenic microbes that we fight.
Hey,
Asking Chat GPT to make an antibiotic, right?
yeah. Can you make an antibiotic? Sure. Where would you like it? We'll see. Airy Ozone wants to know if you know about the new produce coating, Appeal. What's it made
No,
from? I've not heard of that.
no, I'm fascinated because I remember the terrible wax they put on apples when I was a boy. So I'm
Yeah,
going to
yeah.
look into that. That's not one I'll forget. That's good marketing for it anyway.
Kang wants to know, were the first vaccines for viruses? What was the first vaccine for bacteria? So the smallpox was the first, smallpox and rabies, right? Jenner and Pasteur. What was the first bacterial vaccine, Mark? Do you know?
It's got to be from Robert Koch, don't you imagine?
Maybe. Let's look it up.
And yeah, he developed a lot of vaccines for bacteria, again, using attenuated bacteria. It's a lovely question.
Pasteur did a cholera vaccine for chickens.
Okay.
That was in 1872. That was
But again, through attenuation, just trying to grow them at a temperature that is. And it's interesting because clearly evolution was taking place by growing them at different temperatures, and you're making the assumption they've lost pathogenicity.
It was attenuated. Yeah, it was infectious. All right, Frank wants to know, if we removed all the viruses from Earth, do you think humans could survive?
No. And the reason I say that is, and I'm going to use something that I've talked about with bacteria for a long time, is the majority of bacteria that people look at are the ones that have either bad or good interactions with us. So I call them, devil microbes or angel microbes. But the vast majority of bacteria have nothing to do with us. In a similar way, when it comes to viruses, most of our focus is on the things that cause us harm. And this is because we're narcissistic.
Mm-hmm.
But the fact is, every environment you look at has enormous quantities of viruses. And I mean, people say, so some viruses are good. Well, sure, but that's not what I'm saying. I'm saying most viruses could care less. But I promise you that there are some that are doing important and good things. There's a lot of work being done now on the virome that one finds in the gut, and that has to have an effect, and not a negative one necessarily. What do you think, Vincent?
It's funny. I think that it would be a problem for life on Earth to remove all the viruses. Recently, I did a TWIV in Quebec City at an aquatic virus meeting. And one of my guests was a marine biologist from somewhere in California. I can't remember where. I asked him the question: if we removed all the viruses, what would happen? He said the Earth would adapt. He said the Earth is very adaptable. And then after the podcast was over, Curtis Suttle, who was one of the first people to discover viruses in the oceans, he got up and he said, "Vincent, if you ever want someone to say that life would stop after you remove viruses, I'd be happy to do it." So it's obviously a difference of opinion. You can have whatever you want. I think it would disrupt life severely. Yeah.
I've forgotten who it was. It was a very famous microbiologist, and they asked him what would the effect be of full-blown nuclear war. And it's a very famous microbiologist. I'm sorry I don't remember who it is. And this is in the 1960s. He said, life would go on. And they go, what on earth do you mean? And he goes, oh, you mean eukaryotes? Yeah, that'd be a problem.
Yes. Well, right? The eukaryotes survived the the asteroid and killing the dinosaurs, right?
Yeah,
Small
but
mammals
but
survived.
but I mean, it's a really interesting question, and I'm fascinated by the role that viruses play outside of disease.
Of course.
Yeah,
I mean, all the elemental cycles that they participate in, and carbon fixation, and in the oceans, it's incredible. Yeah, I think that would be a big disruptor. So I'm not sure the Earth could recover from that.
I remember John Varley was a science fiction writer who did a number of stories in the 1970s. I don't want to get off on science fiction, but the point is, humans had to leave Earth, and they made sure there were no microbes at all with them.
Mm-hmm.
I know. Hear me out. So the idea was you could do surgery on a tabletop and there'd be no infection. That was his whole point. And it never occurred to him that the vast majority of microbes don't have anything to do with us and yet are very important. And that's the kind of the take home that I make. And that's what makes me a microbial centrist.
Well, notice Frank's question is, do you think humans could survive? So we might not, but perhaps other things would.
Yeah. And considering there's a lot of evidence of the remains of viruses in our genomes, I wonder if they wouldn't just regenerate themselves.
Maybe. We'll,
You
we'll
know,
see when the experiment gets done.
human endogenous retroviruses, they'll activate,
They could,
right?
yeah.
Yeah,
I mean, many of them are mutated, right? But
yeah. They're the rusting evolutionary husks,
that's
right?
right.
But it is interesting to note what percentage of our genomes actually code for proteins. It's less than 2%.
It
So
is.
a lot of our genome is the detritus of I'm going to call it viral movement, for lack of a better term.
One person's detritus is another person's treasure.
Exactly.
You never know. Tona wants to know, does xylitol change the mouth microbes and help prevent tooth decay?
I think there's a lot of evidence that it does affect plaque formation on teeth. It's not a surprise that it would change the microbiome, and I don't mean to get personal with you, but if I take too much xylitol into myself, say from sugar-free candies, there are microbes inside me that have no trouble at all fermenting that, and then I'm very sorry. I don't need to be more open about that.
Mm-hmm.
There's a lot of there are a lot of things that people use. The idea is it's going to change the microbiome that's present, and the idea is that it might cause a little bit of trouble with tooth decay in general. I would just brush my teeth is what I would do.
Okay, John wants to know, what's the importance of microbiology with regard to water bottles? I use 5-gallon exchange at my office to avoid plastic waste. Are there concerns about those?
Concern's a strong word. I think that there's a lot of evidence that there is a robust microbiome that one can find in water bottles. And you might say, well, it's just water. And it is mostly, but there's small amounts of dusts, and there are a whole class of organisms called oligotrophs that live on very, very low levels of nutrients. In addition, these same microbes like to glue themselves to the side of the vessel, forming a biofilm, because that way, as water passes by them, the small amounts of food arrives. And so it wasn't a surprise to me that I could take a student's water bottle and a swab and isolate all kinds of things. And those are just the things that I can grow. There are many, many organisms like Colobacter, Vincent, that really are adapted to that very low nutrient environment. Is that a bad thing? Probably not. When I was dating the woman who's now my wife, and we were living in LA, and one of those big 5-gallon, I was going to show how macho I was to put one of those big 5-gallon things on the crock that she would use to deliver water. I was washing out that crock, and she said, why? It's just water. And I took my finger and I went, ah, and it was like dark brown.
Mm-hmm.
And that was a biofilm. So any microbes can take advantage of so many things. So when I tell students, you know, you're not going to be able to truly sterilize your water bottle. I mean, you could put it exposed to gamma radiation, I suppose, but you're going to find things that survive it. So yeah, wash it with soap and water. That's the best thing to do. but I don't think there'd be very, very big concerns with that. I would certainly keep it away from the light because there are many organisms that can use a small amount of light and fix carbon dioxide in solution. You don't want them growing.
All right. At least one person likes 15 minutes of weekly Racanielling. All right. We'll do it. Doctor.
What is the artwork? You need artwork for this, Vincent.
Yeah. Well, maybe your artist can help me out. All right. Dr. Martin, are there any plants, leaves, etc., which have antimicrobial properties? Garlic, for example.
Absolutely. Garlic is a really, really good example of that, but it has to be fresh garlic because it quickly oxidizes. And there's a wonderful student experiment. You can find it on the internet, finding the antimicrobial effects of the components of salsa. It's a great thing for students to do. And you have these beautiful zones of clearing. But and in fact, there's a lovely quarterly review of biology long form essay about the theory that spices and such are actually antimicrobial. And there are some that are, and some microbes that could care less, but it is absolutely true that garlic is certainly very efficient at it. Allicin is, I think, the name of the compound, but it has to be fresh. And fresh garlic is good.
All right. I have cats that have gingivitis and have wondered if it has anything to do with communal food, water, and passing of microbes. Thoughts?
The hard part with cats is you don't brush their teeth. And they are not eating a diet which is conducive to self-cleaning, like in nature. And cats live longer in captivity than they do in nature. So it's the same issue that you have with dogs. They'll get terrible gingivitis, and you just have to get in there and clean it as best you can. Many veterinarians like the fact that they need to knock out your animal to clean the teeth. I'm sorry, I'm a dog lover. It's a big deal to me. But I think that it's a very different world that they live in with the processed food.
Yeah,
that they do. Mm-hmm.
Yeah, the soft food is not good for.
No.
Yep.
And I can't imagine brushing the teeth of a cat. That would end badly.
It would end badly, yes. Okay, Pete says, if you start from the east side of Dorset and walk west 90 miles along the southwest UK coast path, you travel back through geological time, pre-Jurassic, into the Triassic in Devon, spotting fossils. How cool is that? That's just awesome.
When we were in Ireland last year, on the west coast of Ireland, and went to a wonderful island out there, most of the rock on this island was fossil bearing. And it was so remarkable to see it.
Okay, Andrew says, I understand in the microbial world, if things get tough, drought, lack of nutrients, etc., that prey and predator can get together and cooperate. Is this accurate or just my wishful thinking?
I think it's a lovely bit to consider the snowshoe hare and the lynx, that they do form kind of a biphasic relationship, because if all the snowshoe hares are gone, the lynxes don't have anything to eat, vice versa. So I think that there's a lot of give and take in the environment with this. And I know people have tried to show the same Lotka-Volterra equation, as it's called, with bacterial predators and prey. But remember, when we do that in the lab, we're usually using one prey organism and one predator. But in nature, it's much more complex than
that. Is it important to let my toothbrush dry completely, or should I have two?
I'm more concerned with people that have the toothbrushes out when they flush the toilet, but that's me.
Close the lid, right?
Have you seen the action shots of flushing the toilet, and you get the aerosols all through there?
Yeah. Yep.
I myself don't cycle through. I brush my teeth 3 times a day, and I don't dry it out between each one, but I do change toothbrushes once a week.
John wants to know, are there any interesting ways to make water reasonably potable if stuck in the wilderness besides making a fire and boiling it?
So the only reason I know this is I've had students that wanted to do a project on this, and there's something called a life straw. which is apparently very good at eliminating both protists and bacteria. I don't know that it can do anything about viruses.
Is it a straw that has some UV light in it or something like that?
No, it just has filters
I
built
see.
into it.
Okay. Got it.
Although I had a student who had a water bottle with the UV emitter built into it, and I think they may have been ripped off a little bit on that one.
Mark Martin and Michael Schmidt have upbeat energy. You ever work together?
I have had people confuse me with him more than once.
Well,
I'm the guy with
they've both been on TWIM multiple times together. Yeah, go check out TWIM. Mark is there at least every few months, right, Mark?
Oh yeah, I love being on that show. And the last time we had a lot of fun because I was talking about really wild stuff with geology and microbiology, and everyone got really excited.
Okay, with respect to Ricketts rickettsia, the pathogen of RMSF, PubMed article, lethal effect of RR on its tick vector. The bacteria kills many ticks, leading to a low prevalence of infected ticks in nature. Comment. I'm surprised. I didn't know
Yeah, I hadn't heard that either.
bacteria killed ticks. But some remain and can get into people via a tick bite, right?
That's very interesting. I want to know more about that. Thank you.
Go check it out. PubMed. Can you explain why infections of joint implants are not attacked by our immune system? Dr. Griffin had brought up a study on pus cast this week.
One word, biofilms.
That's it. The microbes make biofilms on the artificial device, and they're very hard to get your antibiotic into, right?
Yeah. And I would think also, I don't know how, gosh, I don't know if there would be invasion by immune cells within a biofilm. I don't know. That's a really interesting thought. I do know that antibiotics don't work very well on infected joints because of biofilms.
Yep, that's absolutely right. Yep. Were tardigrades ever big, like during the Carboniferous? No.
Don't I wish? Everyone wants a shoulder tardigrade right here. There were all different kinds, obviously. The holy grail of tardigrades, by the way, Vincent, is the one that has no mouth parts and lives in a high sulfide environment. So clearly it's chemosymbiotic. And I want that tardigrade. But to answer your question, I've never heard where they were very big because they have the whole issue of lacking a circulatory system. So that limits the number of cells that can make them up. The same reasons that nematodes don't get enormous is just has to do with the ability to transport nutrients to different places. I believe a millimeter is the biggest I've ever heard, which is pretty big.
Vibrio is mostly pathogenic, right? Didn't the split off the non-pathogenic into different genera?
I've never heard that, Vincent. Have you?
No, I mean, are they all pathogenic? Vibrio cholerae are all pathogenic, right?
That's an interesting subject because remember, they're part of their pathogenicity is due to a gene which is carried as a prophage. So if they lack that prophage, that Vibrio cholera, according to 16S and ANI and all the rest, barcoding, would still be called Vibrio cholera, but not able to cause disease.
Right. That's what I'm thinking about. Yeah.
I do want everyone to remember, I once got, a lot of times I will say things and then students go say them in other classes, and then people who aren't quite as fond of me as they might be then decide I'm teaching things inaccurately. And I was once said, is it true that you're saying there are no species in bacteria? And, most people who study microbial phylogeny will say that's true because they're not the way we define it. And the amount of horizontal gene transfer is so enormous. Their family tree is like a kudzu bush. I mean, there's so much gene flow taking place all the time That's where this term pangenome comes from, in fact.
Mm-hmm.
How do you define E. coli? The E. coli that I work with in the lab that was isolated in 1912 at Stanford and been passed in from lab to lab since then is very different from the E. coli that makes you sick at Jack in the Box.
That's right.
And the one at Jack in the Box has 1,000,000 base pairs more in its genome than the E. coli I have in my lab. But by all the standards we use, we both call them E. coli, or
Right.
they're both called E. coli. And that's very different than you see with animals and plants. And I think that's where a lot of this comes from. I mean, if you look at the 16S of Bacillus anthracis and the 16S, the barcodes again, folks, of Bacillus thuringiensis, anthrax versus something that we use as an insecticide, very different types of organisms. That's just one gene.
Yeah. Richard said a failed experiment is still an experiment. Absolutely. Would viruses be better described as shuttles of genetic data?
I think those are mistakes mostly, but they are there. If you look at transduction, for example, or the ability to pick up foreign genes, Vincent, wouldn't you say that that's rare compared to, quote, typical transmission of genetic information?
Yeah. Yeah, I think the transmission doesn't do much for the virus, right? It's an accident. It's
Yeah.
very common, but I don't see that the virus benefits in any way. No.
So when you hear someone say that this antiseptic kills 99.99% of bacteria, that sounds great until you have 10 to the ninth bacteria, right? So these are really, really great questions, by the way. I'm very impressed.
We have a wonderful group here typically on Wednesday night, people who like science. Mark, could evolution put a green fly genome into plants so that aphids emerge with the flower buds?
I so look forward to people, very much like Blade Runner, just like creating different unusual organisms just for fun. Vincent, have I told you this story from my graduate school days? I don't recall. So I had a friend who was in one of the early plant genetic engineering firms. Are you with me?
I'm with you.
And so the exoskeleton of insects is chitin. It's probably the most common polysaccharide on the planet. And that's what makes you go crunch when you step on an insect, right? So if you think about it, there have to be organisms that break down chitin, or we would be this deep in dead insects, right? So the thing that's so cool is that there are lots of bacteria like Serratia that make chitinases that break down chitin. That's good.
Right.
So I knew a fella who cloned the chitinase gene from Serratia and put it in tomato plants.
Mm-hmm.
And you don't have good control, this is pre-CRISPR, of where those genes go. So some events it's being expressed, some events it's not, depends on where it's in. And he had one clone of these tomato plants that was expressing chitinase on the leaves. And as he put it to me, so when an insect tries to chew the leaf, the plant chews back because the mouth parts are made of chitin.
Yeah, that's
and
great.
I said, you can't possibly think you can commercialize this. He goes, no, it'd be cool to do though.
Yeah.
And
that's really good.
Yeah. And that's the kind of ability that we really have. And some people think that that's really scary. And I think it depends on what it is you do.
Yeah.
Right.
Yep. This is a good one. What is the simplest organism that you would call intelligent?
I feel like I'm talking too much, Vincent.
No.
You want to?
No, I mean, you're here to talk. You're here to talk.
How do you define intelligence? I mean, I would argue, it's like the joke with Gandhi. They asked him, what did he think about an intelligent life in the universe? And he says it sounded like a really good idea, right? And
that's good. I love that.
And I don't know how you define it. It's a really tough question. You can absolutely see certain slime molds, Dictyostelium, that can give the appearance of solving a maze for food.
Mm-hmm.
But is that really what's taking place? Is that learning? Is that intelligence? Right? I mean, I don't think anyone doubts that we are, quote, intelligent. Then it comes to the great apes. We don't have any problem with that. Most of us are okay with dogs or cats
Mm-hmm.
or even rats.
Yeah, mammals like raccoons, etc.
Mhm.
Yeah, I think. But what about a snake?
Well, in fact, this I own two reptiles I have in my lab. So baby Carl, the Canyon sand boa, has learned if he boops me on the nose, I'll put him back in his um,
Hmm.
terrarium. So as soon as he's ready, he just goes up and boops me on the nose. So I put him in there. So he's learned that. Is that intelligent? I don't know. You can certainly train flatworms to run a maze. Does that mean that they're intelligent? Right? It's a really tough question how you define it.
Well, you're talking about training, right? That's not really intelligence.
My point exactly.
Yeah,
It's
but I
like
did.
defining life. It's a very difficult thing to define.
But the problem is, we can't really get inside them. We can only see how they're behaving to assess intelligence, and I'm not sure that that's accurate enough.
Right.
Yeah. So reptiles. I mean, fish, right?
Yes.
They seem that they're intelligent. I don't know, but it's not good enough to seem intelligent, right? Although,
Yeah. I have learned it's a really good way to get people angry with you to suggest that some pets aren't intelligent.
yeah, of course. Okay, here's another one. Is soaking strawberries in vinegar enough to clean them before eating? Is it because the acid kills the bad stuff? Mm.
So, are you soaking strawberries in vinegar because of the fruit fly larvae issue that everyone's been talking about on social media? Because if that's the case, I mean, I could make a joke and say it's just extra protein. But the fact of the matter is, there are going to be all kinds of stuff on the food that you eat anyway. And if it's not being cooked, I don't know. I mean, I kind of like a ripe strawberry. I don't know how it would taste in vinegar.
Yeah, I don't know. I use water. Just
Yeah.
I'm okay. I've
Yeah.
been doing it for many years.
But I think that there's a video that's been making the rounds where you put strawberries in something, and from time to time you get tiny Drosophila larvae that emerge from the berry.
Right.
But believe me, you don't want to look carefully at your food.
Okay, let's see. Oh, Dr. Amy brings out the rat and yelling. She pushes all his buttons. She sure do. Oh my gosh.
I've never met her, but she seems to enjoy that aspect of your relationship.
Do you know anything about edible algae?
Yes. Well, there are lots of different. There's the nostoc that used to grow, and I can't pronounce it. That lake in Mexico that the old Aztecs used to eat. Chlorella. I did this as a science project when I was a kiddo. You grew it up and you made it into flour to put into for cooking. And there are certain types of seaweeds that are really delicious. Absolutely, I love it.
Biofilms are 1,000 times more resistant to antimicrobials.
Yep,
Hmm.
yep.
Mark, do
And
you
it
watch?
is.
Sorry,
Go
go ahead.
ahead.
Do you watch the show alone? If so, what behavior of the contestants make you cringe the most microbe-wise? I don't know
So
if you watch that.
I don't watch that show, but I've seen a lot of these survivor type shows and watching people do this. I don't like leftovers. You can imagine I would not do well
Hmm.
under that condition.
There was one here. Let's see here. Teeming with Microbes: A Gardener's Guide to the Soil Food Web by Jeff Lowenfeld. You know this book?
I don't, but now I'm going to go look it up because I love this idea. My graduate work was done in essence with rhizobium that forms root nodules on the roots of leguminous plants and to fix nitrogen. So I love the idea.
Mark, I read if necessary, you can actually make water drinkable by adding small amounts of bleach as long as it's plain hypochlorite with no thickeners, surfactants, perfumes, etc. Is that
Yeah,
right?
that's what they taught. That's what they taught me in Boy Scouts. And I know a lot of, if you'll forgive my saying so, preppers know that. You just have to let the bleach kind of vent off. But you're absolutely right. And honestly, I don't know that someone has ever added microbes to a sample of that water, done that treatment, and monitored what happened. It's a great thing for a student to do as a project, actually.
Yeah, Mark wants an experiment. Yep.
Sure.
Any comments on the seemingly anti-science voices out there? Any personal experience shouting down or being shouted down by anti-science voices? Well, we get that a lot on Twiv. Do you have any experience with that, Mark?
I do. In a long time ago, when I worked in biotech, I felt badly because I wasn't doing research and I wasn't teaching, and it bothered me. And so I thought, what could I do to help? And I would debate animal rights people, and I would debate creationists,
Mm.
and I would do it from a position of trying to be understanding and respectful of different points of view. One's different from me. I dislike confrontation. I don't like fighting, but I feel very passionately about science as a method, as a process. And I found that many people didn't understand what science was. So if I were to call them stupid or duplicitous, that wouldn't change anyone's mind. It wouldn't make me feel better. And so, especially with the creationists, I would try and talk about things because I come from a family of faith. So I'm able to talk about a number of different faiths, actually, because I'm interested in that subject. And the first time that they showed up in an ape mask, for the debate, I realized they weren't serious about having those conversations. So I wished them well, and I exited and I said, this isn't a matter of winning or losing, but when you do what you're doing, you're not interested in talking to one another. We're not learning from one another. And that's was my experience with that. And I mean no disrespect for people that want animals to be treated kindly. I'm one of those people. But when I would encounter people that would insist to me that there's never been any animal research done at all that's benefited humans, I have real trouble with that. And I would tell them so, but not calling them bad names. I said, here's some evidence, here are some things to think about. And you're right, Vincent, it sometimes devolved into ways that made me very uncomfortable, but that's me. Um, I don't want to be the Darwin's bulldog. You know,
Yeah, I mean, I don't like to be disrespectful, although the other side often is. But I try. I think you don't react by being disrespectful and kind. It doesn't work at all. Nobody wins when you do
no,
that.
and it doesn't mean so much of what we do seems to be like creating like getting high fives from our own team
Yeah.
instead of
Yeah.
actually trying to meet people in the middle. And you can't always meet people in the middle. My late mother would, and rest her soul, would say things to me like, I couldn't possibly be of the faith in which I was raised because I believe in evolution. And I would tell her, it's not a matter of believing. I see evidence in my own laboratory from work that I'm doing. And furthermore, I'm the one who was going to church, which my mother never did. So that's what I mean by people sometimes they have not an agenda but kind of an image that they put on things. And I fully admire people that are out there fighting. I think that's good. My approach isn't necessarily the best, but it works for me. That's very dark.
Mark, would you bring home a supermarket rotisserie chicken for family dinner?
I would rather cook it myself.
But if you had to, you would eat that.
Um, whenever I have done that, I usually then cook it. Like we make rotisserie pizzas, which are awesome, by the way. Barbecue chicken pizza
Hmm.
on pitas, it's awesome. I know, Vincent, that's not really pizza. I get it. I understand.
It's okay.
I also
to
eat
each.
pizza with a knife and fork. Can you imagine the trouble I get into about that?
Well, if there's a lot of stuff on it, that's okay. And if you don't, if you've got your laptop there with one hand and you're eating, then the knife and fork works. And yeah, I do
Yeah.
eat and work at the same time, unfortunately, because I have to maximize the time. All right. We have reached the ends of our questions here. I want to thank our moderators tonight. We had Andrew, we had Tom, we had Steph, we had Barb Mack UK, we had Les, and we had Peak Dunning-Kruger, we had Tom. Everybody here, thank you so much for moderating, and thanks everyone for coming. We had about 180 people. We have 176 likes. Thanks, all of you. Next week we will be back, I believe. Let me just check the schedule here. That will be Wednesday. Is it already August next week? Yeah, look at that. July 2nd at 8 p.m. as usual. And it's just me for now, but who knows? I might scare up a guest. And thank you, Mark Martin, for coming on for 2 hours. And you see, it's a lovely crowd here with good questions and a lot of chat. So thanks for coming, Mark.
I really appreciated the opportunity, and I hope that some of you will listen to my own podcast when it becomes available.
I'm sure they will, especially the
And
first
Vincent,
one.
yeah, and well, yeah, you'll be a guest there, Vincent.
Okay, everyone. See you next week. Meanwhile, please stay safe. Good night, everybody.